So again, it's unlikely that the virus is benefiting the organism. There's a specific type of virus that I want to mention, and that's a bacteriophage. And again, this is a specific type of virus, and it is a virus that will infect or will affect only bacteria. So if you said, well, I was just infected by a bacteriophage yesterday, no, you're not, because obviously a bacteriophage only infects bacteria. Now, when we talk about the discovery of a virus, of course, viral particles have been around for a long period of time. But the knowledge of their existence is something that's relatively new. And when they were discovered, their discovery is certainly something that's more recent. So we're looking at something that is not really that old. And one of the first viruses discovered was the tobacco mosaic virus. Its abbreviation, of course, would be TMV. And this is a virus that will affect tobacco plants. And what they know, and it's kind of it's pretty sensible way that they did this discovery. They took a brush essentially, and they saw a plant that was infected with what we now know as TMV, but of course they didn't know it was a virus at the time. They took an infected plant, they took its leaves, and they used a brush to kind of brush the leaves, right? And then they took that tainted brush with the viral particle, unable to see it in a light microscope. You have to think that we were talking about early 1900s, so unable to be seen by a light microscope. Advancements in like heavy-duty electron microscopes were not made at that time. So they took their tainted brush, tainted with they didn't know what, put it on a healthy tobacco plant, and of course, what did they recognize? Well, the tobacco plant that was clean, but you put the tainted particles on it, caused it to become sick. Basically, what they did is they found someone with the flu, had that individual sneeze on their hands, not wash their hands, and then rub another individual's face. Okay, the other person would get the flu, correct? It's the same concept, essentially. Just they were using plants and not people. And it was kind of a unique situation because you were talking about a particle that was smaller than a bacteria but was infectious just like a bacteria can be. So something that is negative but smaller than a very, very small structure. And speaking of their structure, we will spend just a little bit of time going over what viruses are composed of, what's going on with them as far as their inner workings. First off, about their structure, I want to remind us that they are not cells, and they're not made of cells. Like, I wouldn't say that I'm a cell, I would say that I'm made of cells, but either which way, it's, again, there's no way we could include or we could state that virus particles or anything about cells. So that's why I say virus particle and I try not to say virus cell, because it really just doesn't go correctly. So all we are talking about here is having an infectious particle. That's what a virus is, an infectious particle with an outer protein coat. So we've got a protein coat on their outside. Essentially a very, very, very basic structure. Show you this picture real quick, and there you go, a picture of a pretty simple little virus. You'll notice that you've got that purple colored protein coat, a couple of other specialized types of proteins that are on the outside, and you can kind of almost see through this, and you can see that there's really not much going on. And though you might not know about the sizes of stuff, this is extremely small in size, much, much smaller than the cell that would be of your body. So in a microscope, with a light microscope, we can see the inside of a eukaryotic cell. You can barely see this as a dot under the most advanced light microscopes. This right here would be an electron microscope. And you can see that it even still can't zoom in all the way. So obviously very, very small particles. Inside of them, you don't see much, but one of the things you do see inside is you find, or you can see, a nucleic acid. And again, that's inside the particle. And you may find either DNA, and I should have written instead of and, I should write or RNA. You usually could expect to find one or the other going on inside of a cell. Cool? And every once in a while, you can find several important proteins or enzymes I kind of use those interchangeably because, as we know, a lot of enzymes are proteins inside the cell. But again, with this description that you have right there, there's not much going on with a virus. Trying to draw out a picture, you kind of end up with a lot of white space. You've got an area on the inside where you've got nucleic acid, maybe a couple little proteins that I'll throw in right there, maybe a couple specialized proteins like glycoproteins on the outside of the cell, but that's about it. So I'm pointing right there to some specialized types of proteins on the outside, like for instance maybe a glycoprotein. Glyco, of course, means that it's got some type of sugar molecule or sugar piece embedded or next to that protein. Let's see, right there we're talking about having some type of nucleic acid. That, of course, could be DNA or RNA. There's some enzymatic or other small proteins included inside. And, of course, there's your protein coat. I think the thing to really kind of hit home and mention here is that there's not much. There's not much. Very, very basic stuff. Cool? I mean, again, you wouldn't even call this a cell. It's just a little particle, a shell.
So, you're saying not to call it a cell, and it's like on important proteins inside like the particle, is that what you mean? Like inside the particle, something like that?
Yeah, if I said, yeah, see? I should have said particle. Good call. Because you wouldn't even classify this as being a cell. Even though, you associate it as being cellular, because I've drawn a circle with proteins on the outside, proteins on the inside, some type of like DNA or RNA right at this point. You say, well, I've got a cell, but there's so many reasons, and we'll get to a lot of the reasons in just a second why you can't call it a cell. So we'll call it a particle. Again, the only reason that we even include this in the book is because it has an effect on living things. If these particles were just sitting on top of rocks but they didn't affect us at all, we would not include them. But because they can harm you, they become biologically important. The next thing I want to get into is something called taxonomy. Can somebody remind me what taxonomy is? Yeah, basically this idea that we can classify stuff. Now, a lot of people would argue, well, if this is non-living and taxonomy is about classification in biology, how could I ever do taxonomy with these viruses? Or maybe an even bigger question would be, well, why would I be concerned about that? I could understand why it might be important to do taxonomy of plants so I know what plant I have. Well, I think it would be important that you can do taxonomy on a virus because it might help you figure out or determine what virus you have. Obviously, there's a massive difference between having like the virus that causes the flu versus the virus that is known as HIV. Like, obviously, an astounding difference. So you would want to recognize which virus an individual is dealing with. So you have to have some method of taxonomy. Now, usually taxonomy with creatures like a bird or insects or plants is based on what's going on their outside. Their different features, right? Like you can tell a dog versus a cat versus a monkey versus a human apart. You can do morphology. You can study the differences in physical appearance. Harder to do with viruses. One, because they're so small, and two, how different could they be? Right? Because there's so few things that could be different. So our taxonomy for viruses can't be about its look. The only thing that we can do for taxonomy is we could base it on maybe their nucleic acid. In fact, that's what we do. The taxonomy of a virus particle. is going to be based on the type of nucleic acid that is present inside the virus particle. So they're all going to have either DNA or RNA, but they can come in different forms, if you will. Like, for instance, there's double-stranded DNA, like we have. Some virus particles, though, have double-stranded RNA. That's kind of different. Never seen that before, except in a virus. Some viruses use only single-stranded DNA. That's different, too. We always have thought of DNA as being double-stranded, but some viruses have it single-stranded. Some viruses have single-stranded RNA. We've seen that before. And some viruses, awkwardly enough, use single-stranded mRNA as a nucleic acid component. This last group actually is a really special case. You might even want to write their name off to the side. We're talking about the retroviruses here.